students, welcome to EPG Patashala. I am Dr. Sheikh Abdul Hussain, working as scientist at Dairy Technology Division, ICR, NDRI, Karnal. In this session, we discuss the module Effect of Ionizing Radiations on Food Nutrients under the paper Principles of the Food Processing and Preservation. As we know, ionizing radiation is a high energy irradiation which has the ability to break chemical bonds in a molecule. The energy of these ionizing radiations can be transferred to other atomic particle, thus they knock an electron from an atom, creating ions or free radicals. These high energy ionizing radiations can be used to kill microorganisms in food to prevent their spoilage and make them safer, which is the basic concept behind food irradiation. High energy, that is low wavelength radiations such as X-rays, gamma rays and accelerated electrons are the examples of ionizing radiations used for food irradiation processing. During irradiation process, food is exposed to a controlled source of ionizing radiation for the purpose of reduction of microbial load, destruction of pathogens, extension of product shelf life and or disinfestation of produce. Irradiation is often called cold pasteurization because of its remarkable reduction of a number of dangerous microorganisms and because of a negligible loss of nutrients and low degree of sensory changes. Food irradiation is not new. Way back in 1905, patents have been filed in United States and in Great Britain to use irradiation for improving the quality of food. Irradiation of foods must be approved by the US Food and Drug Administration, that is FDA. FDA approval comes only after extensive testing demonstrates that the proposed dose of irradiation effectively eliminates the pathogen or insect of concern and does not generate toxic or carcinogenic chemicals in the food. At present, irradiation has been approved for about 50 different types of food and at least 33 countries are using this technology commercially. However, the toxicity of the chemicals generated and the change in nutritional quality of the food products after irradiation is still a considerable debate. With this, the objective of this session is to make you learn what are ionizing radiations, sources of various ionizing radiations used for food processing, and effect of ionizing radiations on food components. Let us learn what is the mechanism behind microbial inactivation by irradiation process. The reactive ions produced in the foods due to irradiation destroys the microorganism immediately by several means like changing the structure of cell membranes, affecting metabolic enzyme activity, damaging DNA and RNA molecules in cell nucleus. The sensitivity of microorganisms to irradiation varies greatly from microorganism to microorganism and is also dependent on other extrinsic factors. Several factors like size of the organism, type of the organism, number and relative age of the cells in the food, and absence or presence of oxygen affect the microbial destruction in irradiated food. The physical and chemical composition of the food also affects microbial responses to irradiation. Vegetative cells are less resistant to irradiation than spores, whereas yeast and molds have a susceptibility to irradiation similar to that of vegetative cells. Compared to bacteria, viruses are generally require higher radiation doses for inactivation because of their smaller size. Irradiation dose of 5 kg is sufficient to kill most of the spoilage bacteria present in the food. Irradiation dose of 10 kg kills most of the pathogenic bacteria occur in foods. However, some spores forming microbial species, example Clostridium botulinum, and some species capable of repairing damaged DNA rapidly, that is Deinococcus radiodurans, are more resistant to irradiation. Now we will see what are the different sources of ionizing radiation. According to the Codex General Standard for Irradiated Foods, ionizing radiations recommended for use in food processing are 1. Gamma rays produced from the radioisotopes like cobalt 60 and cesium 137. Second, machine sources generated electron beams and X-rays. Gamma rays. Cobalt 60 is produced in a nuclear reactor via neutron bombardment of highly refined cobalt 59 pellets, while cesium 137 is produced as a result of uranium fission. These two isotopes will produce gamma rays, which can be used for the irradiation of food products. Cobalt 60 and cesium 137 emit highly penetrating gamma rays. At present, cobalt 60 is the most extensively employed radioisotope for gamma irradiation of food. Over a period of years, the cobalt 60 or cesium 137 slowly decay to non-radioactive nickel and non-radioactive barium respectively. The half-lives of cesium 137 and cobalt 60 are 30 years and 5.2 years respectively. That is, after these many years or after this period of time, they will be decayed and there will be no further radiation emitted. Second source for the irradiation of food is electron beams and X-rays. Electron beams are produced by electron beam machines which use linear accelerators. Electron beams have limited penetration power and are suitable only for foods of relatively shallow depth. X-rays are produced by bombardment of electron beams with a metallic target. X-rays are more penetrating than gamma rays, but the efficiency of conversion from electrons to X-rays is generally less than 10%, and this has hindered their use for use in food products processing. A major advantage of machine-sourced ionizing radiation is that no radioactive substance is involved in the whole processing system. Hence, these electron beams and X-rays are extensively used for the irradiation of food products. Food irradiation process. During food irradiation process, food product is transferred by a conveyor to an irradiation chamber where the food is exposed to ionizing radiations such as gamma rays or X-rays or electron beams at a controlled rate. The ionizing radiations evenly penetrate the food product and kill the harmful microorganisms, parasites or insects without altering the nature of the food. These radiations do not remain in the food because of their shorter wavelengths and higher frequencies. These gamma rays or X-rays or electron beams, which are used in ionizing irradiation of food, penetrate through the food so rapidly that no heat is produced. Once the food has been irradiated, it must be handled appropriately to prevent recontamination. That is, safe and hygiene handling of the food after ionizing irradiation is very much important. When the radioisotopes, that is cobalt 60 or cesium 137, are used for food irradiation purpose, the irradiation cell, that is the source, consists of cobalt 60 or cesium 137 rods in a stainless steel tubes. These tubes are stored in water and raised into a concrete irradiation chamber to dose the food. That is, no radioactive waste is produced at a food irradiation facility, which is very much important to prevent damage to consumers. Let us learn what is the effect of ionizing radiations on food nutrients. The application of ionizing radiation results in the radiolysis of water, which is a basic component of foods. This triggers the development of species such as hydroxyl ions, hydrated electron and hydrogen ion, which can then induce several chemical reactions with food constituents. Food irradiation can induce several reactions among food components, which include oxidation of metals and ions, oxidation or reduction of carbonyls to hydroxy derivatives, elimination of unsaturation, decrease in aromaticity, hydroxylation of aromatic and heterocyclics, and also generation of free radicals, which can be oxidized to various peroxides. These reactions can initiate lipid oxidation, breakdown of protein components, damage vitamins, and cause undesirable changes in color, odor, and flavor of foods. Quantity of radiolysis products varies as a function of composition of food, as well as with the temperature during the irradiation process and also with the actual dose of irradiation used. Now coming to the changes in lipids caused by irradiation, lipids are less stable food components and hence very sensitive to ionizing radiation. Lipids undergo both oxidative and non-oxidative changes during exposure to ionizing radiations. Exposure of fats to ionizing radiation may induce many auto-oxidizing and hydrolytic reactions leading to undesired organoleptic changes and losses of essential fatty acids. The range and the nature of changes caused by ionizing radiation depend on the type of fat and its content of unsaturated fatty acids. Moreover, irradiated animal fat is more resistant against auto-oxidizing processes compared to plant fats. Let us learn how irradiation doses are expressed and classified. Now we will see how the irradiation treatment given to food product is measured and how it is classified. Since we are treating food with some irradiation source, we must measure the intensity of the dose so that we can effectively classify the different doses of irradiation. Generally, the amount of radiation that a food product absorbs is measured by a dosimeter. The basic unit for measuring irradiation dose is Gray. Gray is the amount of irradiation energy that 1 kilogram of food receives. The irradiation dose that is applied to a food product is expressed in terms of kilogray. It is equivalent to 1000 grays. In 1980, the Food and Agriculture Organization of the United Nations, the International Atomic Energy Agency, and the World Health Organization combinedly stated that foods irradiated up to an overall average dose of 1 megarad, that is 10 kilogray, presents no toxicological hazard and introduces no special nutritional or microbiological changes. Hence, they can be considered as safe. That is, food products which have been treated up to 1 megarad or 10 kilogray are generally considered as safe by these three agencies. The irradiation dose applied to a food product will depend upon the composition of the food, the degree of perishability, and the potential to harbor harmful microorganisms. That is, the more the potential of a food to harbor harmful microorganisms, the more irradiation dose has to be given to that food product. Now we will see how the irradiation doses have been classified. Irradiation doses applied to food products have been classified into three main categories. One is radiorization, another is radicidation, and the third one is rad-apartization. Radiorization is a low dose which is applied up to 1 kilogray. Generally, this radiorization is used to kill parasites, insects, and larvae after harvest, and also to inhibit sprouting and slow ripening. Radiorization is generally applied for food products like fruits, vegetables, wheat flour, potatoes, garlic, onions, and some non-citrus fruits. The second dose is radicidation. It is a medium dose applied up to 1 to 10 kilogray. Generally, this radicidation is applied as a pasteurization treatment to eliminate spoilage organisms and pathogens. Food products like strawberries, grapes, fresh or frozen seafood, and poultry and meat will be irradiated using this radicidation. The third dose is rad-apartization. This is a high dose which is applied up to 10 to 50 kilogray. Generally, this rad-apartization is applied to sterilize food for immunocompromised people and also to eliminate some viruses from food products. It is also used to decontaminate some food additives and ingredients. The applications of this rad-apartization is also in pathogen-free hospital foods and also to sterilize spices, enzyme preparations, gums, and some aromatic substances. Let us see the changes occurring proteins due to irradiation. Free radicals formed by the irradiation of water catalyze the changes in proteins. Damage caused to protein by ionizing radiation include deamination, decarboxylation, reduction of disulfide linkages, oxidation of sulfhydryl groups, breakage of peptide bonds, and changes of valency states of the coordinated metal ions in enzymes. The products formed after irradiation of protein include carbonyl groups, ammonia, free amino acids, hydrogen peroxide, organic peroxides, and etc. At high doses of irradiation, cross-linking can occur between free amino acids and protein or proteins and proteins, leading to the formation of new proteins. Cross-linking between protein and lipid molecules can also occur during irradiation. Changes brought about by irradiation affect the primary, secondary, and tertiary structures of proteins. These changes in the structure of protein is ascribed to the reduction of the sulfur-sulfur bond or the oxidation of the sulfhydryl group caused by irradiation. In the absence of oxygen, decarboxylation, dehydration, and polymerization are the main changes observed in lipids subjected to irradiation. Radiolytic products obtained in this case include CO2, CO, H2, hydrocarbons, and aldehydes. Fatty acids undergo preferential cleavage in the ester carbonyl region, giving rise to certain radiolytic compounds that are specific for each fatty acids. Alkanes and alkenes are the most abundant, representing 95% of the volatile substances formed by lipid radiolysis. During irradiation, fatty acids undergo preferential cleavage in the ester carbonyl region, giving rise to certain radiolytic compounds that are specific for each fatty acids. Alkanes and alkenes are the most abundant, representing 95% of the volatile substances formed by lipid radiolysis. The amount of radiolytic products generated depends on the irradiation dose and are generally small in quantity. Free fatty acids also will be released from the cleavage of neutral lipids or phospholipids after irradiation treatment. Free fatty acids are not harmful, but they cause a slight reduction in absorbability of long chain free fatty acids like palmitic acid, stearic acid, etc. Irradiated fatty acids are digested and absorbed at a slow rate than non-irradiated fatty acids, but there is no alteration in their nutritive value. However, after several extensive studies, it can be concluded that when lipids are irradiated under conditions anticipated for commercial food processing, it does not result in significant loss of nutritional value. Amino acids inside a protein are less liable to irradiation than free amino acids. Sulfur-containing amino acids like cysteine, methionine, and tryptophan, along with aromatic amino acids, are the most susceptible to irradiation damage. For aromatic amino acids, the indole ring of the aromatic group is the primary target of oxygen radicals. When sulfur compounds are submitted to radiation in the absence of oxygen, hydrogen sulfide and sulfide are formed in large amounts. In the presence of oxygen, the amount of ammonia and sulfuric acid produced increases. The typical order of irradiated foods is related to the formation of these sulfuric compounds. However, data obtained from literature indicate that irradiation at commercial doses, that is 2 to 7 kg, has no significant effect on the nutritional value of proteins or amino acids. Now we will see how ionizing radiations affect saccharides or carbohydrates in food products. Ionizing radiation affect saccharides when they are either in a solid state or in a solution. Saccharides are extremely sensitive to radiation in the crystalline state, and the radiolytic products obtained will depend on crystalline form of saccharides as well as the content of water present in it. When saccharides in solid state are irradiated, their melting point may be reduced and also their optical rotation changes. When saccharides in aqueous solution are exposed to irradiation, both the optical rotation and the index of diffraction may be reduced. The irradiation of aqueous monosaccharide solutions gives rise to hydrogen, CO, CO2, formaldehyde, malonaldehyde, glyoxal, aldonic acid, uronic acid, sugar polymers, and deoxy compounds. Some of these substances, if produced in higher concentrations, they could present a carcinogenic risk. The content of carboxyl acids may increases if oxygen is present during irradiation of saccharides. The fission of the glycosidic bond may also occur during the irradiation of saccharides. Irradiation of polysaccharides, for example, starch, leads to reduction in viscosity of solution, and at a higher doses the gel does not form. Starch grains become fragile and disintegrate, and also starch exhibits an increased reactivity to alpha amylase after irradiation treatment. During irradiation of saccharide rich foods, a small amount of potentially hazardous substances, for example formaldehyde, malomaldehyde and deoxysaccharides, which are harmful to human health, may be formed. Now we will see how the irradiation dose affects the vitamins present in food products. Effect of ionizing radiations on vitamins is influenced by the medium in which the vitamins are present. Fat soluble vitamins are affected by radicals produced during fat irradiation. Similarly, water soluble vitamins are affected by radical formed by water irradiation. In the case of fat soluble vitamins, the free radical mediated reactions are negligible since they will mostly recombine with positive lipid ions. Since vitamins are in quite low amounts in most foods, the primary hydroxyl radicals will mostly react with other major food components like lipids, proteins and carbohydrates before they are going to react with the vitamins. The vitamins are thus more affected by the secondary radicals formed by the interactions with the major components, which are mostly hydroperoxides. Among the fat soluble vitamins, vitamin E is the most irradiation sensitive, while vitamin D and vitamin K are highly stable. At high irradiation doses, vitamin K may be lost significantly. Among the water soluble vitamins, thiamine is the most irradiation labile vitamin. With increasing temperature during the irradiation processing, thiamine destruction will be increased. Folic acid is sensitive to irradiation at a dose of more than 25 kg. Riboflavin, niacin, biotin, cobalamin, choline and pantothenic acid are relatively stable to irradiation. This slide shows the relative radiation sensitivity of vitamins. Among the fat soluble vitamins, vitamin E is the most sensitive, followed by carotene, vitamin A, vitamin D and vitamin K. While among the water soluble vitamins, vitamin B1 is the more sensitive, followed by vitamin C, vitamin B6, vitamin B2, folate, niacin and vitamin B12. Now we will see how irradiation dose affects minerals and trace elements present in food products. Irradiation did not significantly affect the bioavailability of the minerals or trace elements present in the food products. Now coming to the organoleptic changes which are affected by the irradiation of food products. Textural alterations and development of off flavors are not significant when food is irradiated at lower doses than 2 kg. The changes in foods at lower doses of irradiation are similar to those occurred during heat treatment equal to pasteurization. At higher doses, that is more than 5 kg, irradiation leads to off flavors and some textural alteration. Several methods, namely exclusion of oxygen from the package, replacement of oxygen with inert gas, and addition of antioxidants, etc., can be employed to decrease the detrimental effects of irradiation on food products. FDA has established certain regulations for labeling of irradiated foods. Labels of irradiated foods must contain the words treated with radiation or treated by irradiation, and the packaging label of irradiated foods must also display the irradiation logo, which is called the radura. Here it is shown in this figure. However, no label is required for food products that contain irradiated ingredients such as spices, etc., as long as the entire product has not been irradiated. That is, if a food product or package contains some irradiated ingredients, it need not to be written on the labeling as treated with irradiation or treated by irradiation. So dear students, let us summarize this session. Ionizing radiations are high energy irradiations which has the ability to break chemical bonds in a molecule. These ionizing radiations such as X-rays, gamma rays and accelerated electrons can be used for food processing. Irradiation destroys the microorganism by altering the cell membranes and also by altering the metabolic enzyme activity and also by damaging DNA and RNA molecules in cell nucleus. Among the food constituents, animal fat is more resistant against irradiation compared to plant fats. Especially polyunsaturated fatty acids are more sensitive to irradiation. Saccharides are extremely sensitive to radiation when they are in crystalline state. At higher doses, that is at more than 5 kg, irradiation leads to off flavors and textural alterations in food products. FDA has established regulations for labeling of irradiated foods. That is, irradiated food must contain irradiated word on their labels and also should bear the symbol radura. Thank you.